I was just saying, for technical reasons, Azza Ahmed is not going to be able to present today. So we're just having one speaker, who is Yosr Hamdi. She's working at the Université Laval in Quebec and also in Pasteur. Yes. Okay, so I think Yosr, you can
Sorry.
Thank you very much for being our speaker today.
كيف؟ J'ai pas compris. Est-ce que je dois commencer?
Yes, you're welcome.
Oh, okay. Thank you very much for the invitation. I will start the presentation just in a few seconds. I have to share.
Anmol, can you activate your camera, please? Because it's...
Okay, just 1 second. Ça marche.
Anmol, can you please switch off your webcam? Anmol, please. Turn off. Okay, sorry. Thanks. Okay, Yosr, you have the floor.
Yeah, just 1 second. Pour la faire. Non? En principe, ça marche, mais juste, je n'arrive pas à mettre ça en plein écran. Ouais. Voilà. Là que ça marche. So now it works?
Yes, maybe you're not going to be able to put it in full screen. We already had this problem last time. So even if it's not full screen, try your best to enlarge it in the window, but don't switch to full screen mode. Just make your window as large as possible, or maybe hide the toolbars. Hide as many toolbars as possible. Try to hide the menu bars.
The menu bars and you
can close the animation panel on the right. Just close it, and close the thumbnails on the left, maybe.
Ça marchait quand on a essayé l'écran.
Est-ce que je continue?
Okay, I will start. So I want just to be sure that you are hearing me. Je m'entends, c'est normal. Okay, so comment ça marche؟ نسمع في روحي normal. Okay, so I'm Yosr Hamdi from Institute Pasteur de Tunis. Today we will speak about breast cancer and the major challenge of genetics in African population from association to function. Just I want to be sure that you are hearing me. It's okay? Okay. So breast cancer is one of the top cancers in women worldwide, and it's increasing, particularly in developing countries where the majority of cases are diagnosed in late stages. So it's the most common cancer among females, with 1.6 million of cases per year, and is also considered as the most common cause of cancer death among females, with 14.7% of cancer death is caused by breast cancer. So here we have some facts, and we have the incidence rates in blue and mortality rates in red, and we have to compare. We will compare these rates in North America and in North Africa. So as we can see here, we have the incidence rate in North America. Breast cancer incidence rate in North America is 2.5 folds more than in North Africa, but the mortality rates are 1.2 folds less in North America than in North Africa. So here we have a problem between countries. So breast cancer risk factors, we have for this disease multifactorial disease. So we have some demographic factors, we have some hormonal factors involved in this cancer, environmental and lifestyle factors, and we have also some physical characteristics that are associated with breast cancer, such as obesity in postmenopausal women and breast density. But one of the most studied and known breast cancer risk factor is family history of breast and ovarian cancer. When we speak about about family history, so we speak about the genetic effect of some variants associated with breast cancer. In this figure, we have, in this axis, we have the allele frequency, and in this one, we have the effect size. So in breast cancer, we have three main categories of variants associated with breast cancer. We have rare alleles that have a rare frequency, but they have high effect size. We have low frequency variants that have intermediate effect size. And the third one is common variants that confer a low or a modest effect size. So today we have 50% of the genetic component of breast cancer is explained what we have here. 50% of this breast cancer genetic component is explained by a combination of high and intermediate and low risk effect variants. As we can see here, we have BRCA1 and BRCA2 that are the two genes most involved in breast cancer. And here we have the part of the polymorphisms or the SNPs that are associated with breast cancer component. So now I will show you some of the work that I have done in the research center of Laval University at Quebec in the laboratory of Professor Jacques Simard. So what we have done, we participated in the COGS project, which is the Collaborative Oncological Gene Environmental Study, that studied the association of some polymorphisms with breast cancer, ovarian cancer, and prostate cancer. So we used an Illumina Infinium custom array, and we genotyped 220,000 SNPs in almost 200,000 samples, of which we have 95,000 samples that have breast cancer, and that belong to the BCAC consortium. The BCAC consortium is the Breast Cancer Association Consortium that was formed in 2005 at the University of Cambridge, and is considered as one of the largest, it's the largest breast cancer consortium worldwide. And it globes Caucasians, Asians, Afro-Americans, and they have they identified the majority of the breast cancer associated variants. So the work that we have done with the BCAC consortium or the COGS project, we have identified two categories of variants. We have variants that have rare, high risk, and coding variants, and we have this category of common, low risk, and non-coding or regulatory variants that we identified by genome-wide association studies. The main goal of the work that we have done is to integrate breast cancer genetic discoveries into clinical practices and public health programs. So including them in genetic testing, such as molecular diagnosis, improving cancer risk assessment, and enabling risk stratification based on each woman's personal genetic characteristics. So we have done a genome-wide association study, and we have also done a genome-wide functional study in order to identify more variants associated with breast cancer in the Caucasian population. So the work that I'm presenting here is the work that I've done in Quebec in the Laval University. So then we will switch to the other project that I've done in the Institut Pasteur of Tunis. But now I'll present just the work that I've done in Quebec. Well, these two genome-wide studies, the association and the functional studies, we worked on the functional genomics, and as we can see here, we have the genome. And the biggest part of the genome is represented by no-coding regions. We have also 90% of the genome-wide association study hits are found in non-coding regions. So we tried to study the relationship between regulatory SNPs and disease risk. So regulatory SNPs or R-SNPs, they can increase or decrease gene expression, what can influence on the metabolic pathway, and this can be associated, the R-SNPs, that way they can be associated with the disease risk. So our hypothesis was to see how much functional regulatory polymorphism can be associated with breast cancer risk. We started with a genome-wide functional study, so a genome-wide differential allelic expression study. What we mean by differential allelic expression or allelic imbalance is here. So as we can see here in the DNA level, we have two alleles, C and T. When they are transcribed into mRNA, we expect to get as C's than T's, so we have the ratio of 1. But sometimes we can observe more T's than C's, as we have the example here. So this is what we call an equal allele expression or allelic imbalance. So what we have done, so we identified all the SNPs that are associated with allelic imbalance on the genome. And then we picked up those that are on breast cancer candidate genes, and we submitted these SNPs to the COGS list. So we identified all the SNPs on the genes that candidate genes for breast cancer, and we studied the association of these polymorphisms with breast cancer risk. And one of the results that we have, so we identified a new breast cancer association locus on the chromosome 4 and on the gene that's called Abraxas. So briefly, just to describe the role of Abraxas here, Abraxas is directly linked to the BRCA1 gene in the DNA repair pathway. So Abraxas, when it's associated with BRCA1, they can repair the DNA damage caused on the DNA. Well, now the challenge is to find the causal SNP, so the functional SNP that is associated with breast cancer on this locus, on the 4 chromosome for Abraxas. Here we have Abraxas, and I have the animation, so it does work like that. But I have to show the SNPs that I have. Well, anyways, just a technical problem, because I have just under this, we have the list of the SNPs that is associated with the breast cancer. So all of the SNPs that I have here are SNPs that in perfect linkage disequilibrium. So the R squared is of 1. When we have one SNP that is associated with the breast cancer, so all of the other SNPs are also associated with the disease. then the problem is how to identify the causal SNP, the functional SNP that is associated with the disease. So we have done all this work, and we submitted the paper on PLOS Genetics that has been accepted. So this is an example. The work that I presented for 4Q21 is an example of how to search for causal SNPs on the identified breast cancer associated variants. So now I will switch to the current study that we have on breast cancer in the Institute Pasteur and the laboratory of Professor Sonia Abdelhak. The work is entitled Functional Annotation of All Known Breast Cancer Associated Loci. So it's a kind of a genome-wide haplotype study in the general Tunisian population. So I'll start by describing a little bit the North African region. So, you know, I guess that you know, all of you, that the North African region is composed of 5 countries just here, and is found in a strategic position with interaction with Africa, with Europe, and with Asia. We also have a very interesting population history on North Africa with different civilizations that passed by this region. So the genetic wealth of the North African population is due to the endogamy, consanguinity, to the heterogeneity due to geographic position, heterogeneity due to the historical migration waves, but the problem is that the North African populations are not studied by the HAPMAP project, neither by the 1000 Genomes project, neither by any other project. So we have to do all the work by ourselves. And why it's important to study such a heterogeneous population is the same thing for other low LD populations or for other African populations. It's very useful in population genetics in order to identify the causative SNP. So as the example that I have here, for the population 1, the tagging SNP can be on the same haplotype as the causative SNP. But in the second population, the tagging SNP, we can find it in one haplotype, and the second causative SNP can be found on another haplotype. So it's interesting to compare haplotype constructions between populations in order to identify causative SNPs. So in order to study the genetic architecture of the general Tunisian population, the breast cancer, we use the data from a genome-wide study on a chip of Affymetrix 6.0. So we extracted genotypes of 135 Tunisians representing the general population, and we studied almost 90 known breast cancer associated loci. So as we can see here, we have our breast cancer locus. We identified all the SNPs that are genotyped on the Affy 6.0, and also identified SNPs that are on 50 kb upstream and downstream of each locus in order to identify the regulatory regions or to study the regulatory regions on each locus. So a total of almost 1,600 SNPs that have been studied on the Affymetrix 6.0, and all these SNPs are found on different chromosomes and in different loci associated with breast cancer. What we have done is we extracted genotypes for these healthy Tunisian subjects. We identified the minor allele frequency in the Tunisian population. We phased haplotypes. We identified LG plugs for all loci. We compared the data that we found with other populations from HAPMAP project and for other projects in order to see if we can identify some causal variant associated with breast cancer. So I will show you just an example of BRCA1 that we studied here. What I've done is I combined knowledge for variants associated with breast cancer in the Tunisian population, as we can see here, and data from association in the Caucasian population. So for these areas is associated with breast cancer risk in Tunisia and in the Caucasian population. So one of the interesting SNPs that we worked on is this one, which is considered as a haplogroup tagging SNP. So it's not just tagging one haplotype, it's tagging a group of haplotypes, as we can see here. So it's one of the interesting SNPs that we worked on. And this SNPs, we already published work on this SNPs and this association with breast cancer in the Caucasian population. So interestingly, we found when we have done this study on the general Tunisian population, we worked on the LG block carrying breast cancer gene, and we found another polymorphism, which this one, that it seems that in a perfect LD with the haplogroup tagging SNP. So this RS, it seems a very interesting one because it may be a causal SNP. So we've done some functional work, and we found that the RS that we genotyped on our chip is associated with allelic imbalance, is associated with a QTL, as we can see here, some results that we have extracted from the gene var. So it's maybe the causal variant and the functional variant that is associated with breast cancer. Then we have done some genetic, some comparison between populations. We extracted the data for the Tunisian population and for other populations of the BRCA1 LG blocks in order to compare between population. And as I said at the beginning, so when we compare data from different populations, we can identify if the causal SNP that we identified as functional is the one associated with breast cancer or not. So with this, so the BRCA1 variant that I presented is just one of the other polymorphisms that and variations that I studied in this current study. So what we have done, we identified the frequency of at-risk haplotypes, the frequency of heterozygous haplotypes and homozygous wild-type haplotypes in the Tunisian population. It's very interesting because we can see here some differences between the frequency of some loci, such as the 4Q here and the 22Q here, compared with the 11Q and the 6Q that we have just here. Okay, then before was one of the, I think that's one of the last slides that I have. I've done again a PCA, so the principal component analysis in order to compare the breast cancer genetic architecture in the Tunisian population compared with Europeans and with Africans. And as we can see here, it seems that the Tunisian population is intermediate between Europeans and Africans, but it's more close to Europeans than to Africa. Here we have another PCA where I added some populations from North Africa and from Europe in order to see where we can see the Tunisian population, the North African population just here in blue, and the European population. So the conclusion is that the breast cancer architecture in the Tunisian and in North African population it seems to be as intermediate between Europe and Africa, but it's more close to the European populations than to African population. Well, finally, to the takeaway message, so haplotypes in North Africa are not diverse enough to be African. So when we constructed haplotypes and when we compared LG blocks, when we see the PCA results, the conclusion is that haplotypes in North African population are not diverse enough to be African and not sufficiently homogeneous to be European. So just intermediate enough to be suitable to overcome the major challenge of molecular genetics in African population. The other results that we have here, we identified a new causal variant on the BRCA1 gene that seems to be in LD with a haplogroup tagging SNP, and this can be used in in the genetic testing or the genetic diagnosis. So we are trying to develop a new breast cancer genetic diagnosis tool using our results, the results that we identified just here. And so finally, we are working in order to establish a consortium, North African Breast Cancer Consortium, as we can see here. So we will work on different aspects on genetic demography studies, on breast cancer susceptibility genes. We will also work on psychological aspects and ethical and social and legal perspectives. So to finish, I want just to thank Dr. Jacques Simar's group in Canada, Professor Sonia Abdelhaq in Institut Pasteur de Tunis, Professor Samir Boubakar in Institut Pasteur de Tunis, and all our collaborators. So thank you very much. Yes. Ok. Qu'est-ce que je dois arrêter le partage? I'm hearing, no, I'm just here. There's some questions. Okay. Yeah, yeah. Thank you.
Investigate. Really. No, I just worked. I'm just to come back to the PCA that I showed. So we worked on the Tunisian population. Then we compared our results with 2 different European populations and 4 different sub-Saharan African populations. So the results that we found here is that the Tunisian population, it seems to be closer to the 2 European populations than to African population. But when we speak about North African populations, the other North African populations, are you the slide of the PC? Are you seeing that slide? Yeah, yeah. Okay. So the populations, the North African population here, just in blue, are again more closer to these European populations than to African. So the question, this data we have for breast cancer loci, it's based on the frequency of polymorphisms associated with the breast cancer, known to be associated with breast cancer. When we compare, Tunisia is more closer to Europe and Mediterranean countries than to sub-Saharan African. So I don't know if I answered to the question or not, but For the other populations, such as Mali or Chad or Sudan, we don't have data for these populations about breast cancer loci. I think that no studies on these polymorphisms have been done for these populations, so we cannot compare these populations with our population or with others. What I showed here is just the populations where that we have data for breast cancer genetic variations. Mm-hmm. Mm-hmm. Yeah. Yeah, okay. But also perhaps if we study these populations such as Chad, Mali, those who are near to our population, perhaps they will be close to the Tunisian population. But as I said, really, I don't have data for this. Yeah. Okay. Other questions?
Yeah, it's closely related to Victor's question. I mean, or maybe it's just a reformulation. It seems that you're exploring only a subset of genomic locations that are known to be risk factors for breast cancer.
Yeah.
In the other populations or the populations in the 1,000 Genomes Project.
Yeah.
But you're probably missing some very important genomic positions that maybe are risk factors for the Tunisian populations, for maybe the
Exactly.
African populations, and that are
Exactly.
not risk factors for other populations. So are you thinking about maybe the idea of starting from scratch and exploring the whole genome and see, okay, what are the risk factors that may be specific to North African populations?
Okay, so I want just to clarify some points. Here, what we studied, we decided to start by studying our healthy population, or what we call the general population, before working on our own patients with breast cancer. But in parallel with this study, we are doing NGS, we are doing whole exome sequencing on Tunisian patients with breast cancer. And we identified some results for, as you said, for variants that are specific to our population. But we cannot merge this data with this, because what I present here is a healthy population, is from the general population. And the other variants that we identified, and we are confirming these results right now. So there are other mutations that are specific to the Tunisian populations. So we don't have data for other populations for these variants, and so we cannot use this data to compare between Tunisia and other population. But it's a very interesting point that you here, so what you said. So because we have data about the variants that are specific to our population. We studied this. We have done many whole exome sequencing, and yeah. But to compare with other populations, we cannot do that because these variants are not found in other populations. Yeah. Mm-hmm. Yeah. Yeah, that's a very interesting question. And what I have here is the frequency of haplotypes in red, haplotypes, oops, at-risk haplotypes for different loci in the general Tunisia population. So as we can see, for example, for, I don't know, for the FGFR2 locus, the general Tunisian population is at 22%. 22% of the general Tunisian population have this, are carrying the at-risk haplotypes. So that means that probably a lot of these people will develop cancer. So it's really very important to choose, and how I think that it's a big problem, not just for our population, but when we work on breast cancer research or another cancer research or complex disease research. So it's very important to choose our controls. First, in question of age, because at a specific age, we are sure that the patients, if it didn't develop the disease at this age, for example, at 50 years old, they will not develop the disease after that age. So we have to take many points into consideration: age, results of diagnosis, results of controls, such as mammography controls for our control subjects, and many considerations. But ideally, we have to genotype some of the breast cancer associated loci to see if our controls are carrying the at-risk locus or not. It's the ideal way to choose our, yeah. Okay.
Okay, thanks. It seems some people could not hear Victor's question, although Yosr and myself could, so I tried to fix this problem. Do we have other questions? If I may, I would like you to elaborate a little bit on the first part of your talk when you talked about allelic imbalance and the role of allelic imbalance in your study on this. Can you tell us a bit more, and basically what did you sequence, or did you separately did you have markers on the two alleles of a chromosome, or?
Okay, I will just try to return back to the slide where I'm not understand, because it should be CPG. Excuse me, just 1 minute to fix this problem. Si je vais monter en rouge. J'ai la présentation. Okay. Sorry, just 1 second. So for the allelic imbalance stuff here, just here. So when we participated into the COGS projects just here, so the COGS project, it was on different cancers and on different consortia. The total number of polymorphisms that were submitted to the chip of COGS is 220,000 SNPs. So among these SNPs, we decided to submit SNPs, functional SNPs. Our hypothesis was to see how much functional SNPs can be, functional SNPs associated with allelic imbalance, how much they can be associated with breast cancer risk. So the SNPs that we submitted to the COGS chip are SNPs associated with allelic imbalance. The allelic imbalance is just when we have two alleles, and when we have one allele that is more expressed than another. So that means that this allele can be more expressed, and when it's associated with the disease, so that means that the disease that will be causally associated with the disease among the via the gene expression level. So from this study, it was a genome-wide allelic imbalance study, we choose some SNPs that are on breast cancer candidate genes. These SNPs were submitted to the COGS list, and we studied the association with breast cancer risk for the selected SNPs that are associated with allelic imbalance. So we tried to see if there's any association of the functional variants and breast cancer loci and breast cancer risk. And from the study, we identified the Abraxas gene, which is a new breast cancer associated locus. So the variants on Abraxas are already known to be associated with allelic imbalance. So here we have data for association and for function for the submitted SNP. So was it clear, the answer?
Yes. Yeah. Thanks.
But if you have any other questions on the allelic imbalance, I can.
It means in your sample you clearly know you have markers on chromosomes, so you clearly know whether your two chromosomes are the two chromosomes of the same individual, or you just don't care of the. You just don't see at the individual level. You're just sampling genomic positions without caring of whether it's on.
Yeah, exactly. So different polymorphisms in different genomic regions associated with allelic imbalance were submitted in order to see how much this polymorphism can be associated with breast cancer risk. This was the idea.
Okay.
Okay.
I don't know if it's the case for everybody, but my screen is black, so I don't know if it comes from your side. Are you in full screen or something?
Okay, sorry,
Okay.
we are just trying to fix this problem. قال الإكرام تاعو نوار. كيف ما فهمت. قال الإكرام عنده black.
Okay, so long. Yeah, thanks. So long. Do we have other questions from the audience?
أما بكري وهو ما يمشيش.
So, is there other questions?
I see no question on Twitter. But remember, also the people on YouTube see this with some lag, some delay.
There is no comment on YouTube channel.
Okay, so maybe we can thank you once more, Yosr, and for being available and for also accepting to present with a very short notice, because we only contacted you a few days ago. So thanks again.
No problem. No problem. Okay, thank you very much for the invitation, and so have a nice day.
It was our pleasure. Thank you so much. Thanks to the audience. And so we'll see you all for the next seminar. Is probably it will take place probably next month, end of next month.
Okay. Thank you.
Okay. Goodbye, everybody.
Bye.
Thank you. Bye.
Bye-bye.
Sorry for the jump traffic.